When we think about powerful disease-fighting nutrients, our minds often jump to vitamins, antioxidants, or exotic superfoods. But one of the most potent protectors against colon cancer is something much simpler, far more accessible, and often overlooked: dietary fiber. Fiber isn't a single substance. It's a family of plant-based carbohydrates that our bodies can't digest in the traditional sense. Unlike sugars and starches, fiber passes through the stomach and small intestine largely intact. But here's where the magic happens. Once fiber reaches the colon, it becomes the favorite fuel of trillions of bacteria that live inside you. These bacteria, collectively known as your gut microbiome, aren't just passive residents. They are active partners in your health. When you feed them the right kind of fiber, they transform it into compounds that have remarkable anti-cancer properties. Among the most important of these compounds are short-chain fatty acids, particularly one called butyrate. Butyrate is a small molecule with a big role. It serves as the primary energy source for the cells lining your colon. This might sound simple, but it's actually critical, because when your colon cells are well fed with butyrate, they function optimally, maintain a healthy structure, and resist abnormal changes that can lead to cancer. Research has shown that butyrate doesn't just nourish healthy cells. It also has the ability to signal cancerous or precancerous cells to shut down their growth and even self-destruct through a process called apoptosis. This means that something as unassuming as eating more beans or whole grains could directly influence whether potentially dangerous cells in your colon survive or die. The protective power of fiber extends beyond feeding your microbiome. High-fiber foods also physically alter the environment in your colon. Fiber adds bulk to your stool, which speeds up the transit time of waste through your digestive tract. This is important because the longer waste sits in your colon, the more time any harmful substances, including carcinogens, have to interact with your colon lining. By moving things along more efficiently, fiber reduces that exposure window. In addition, fiber helps bind to certain harmful compounds, including excess bile acids, and carries them out of the body before they can cause damage. Think of it as a cleaning crew that works around the clock to sweep away potential threats. What's fascinating is that not all fiber works in the same way. There are two main types: soluble and insoluble. Soluble fiber dissolves in water to form a gel-like substance, slowing digestion and providing steady nourishment for beneficial gut bacteria. It's found in foods like oats, beans, apples, and carrots. Insoluble fiber, on the other hand, doesn't dissolve. It acts like nature's broom, adding bulk and helping to keep things moving. This type is abundant in whole wheat, nuts, seeds, and many vegetables. For optimal colon health, you need both types working together. Soluble fiber nurtures your microbiome, while insoluble fiber maintains a healthy flow through your digestive system. When you combine them in your diet, you create a two-pronged defense system: biochemical protection from short-chain fatty acids and mechanical protection from reduced carcinogen contact. Modern dietary patterns, unfortunately, have created the perfect storm for colon cancer risk. Highly processed foods, refined grains, and low fruit and vegetable intake have left many people consuming far less fiber than their bodies evolved to thrive on. In fact, in many Western countries, the average person consumes barely half the recommended daily intake of fiber. This fiber gap leaves the gut microbiome starved of its preferred fuel, leading to reduced butyrate production, weakened colon cell defenses, and a higher risk of inflammation, all conditions that create fertile ground for cancerous changes. Chronic inflammation, in particular, is a key driver in the development of colon tumors, and diets low in fiber often correlate with higher levels of inflammatory markers in the blood. Emerging research has even shown that certain types of colon cancer respond differently depending on fiber intake. Populations with naturally high fiber diets, such as those based on traditional plant-heavy cuisines in parts of Africa and Asia, tend to have dramatically lower rates of colon cancer compared to populations consuming Western-style diets. In some studies, when participants switched from a low-fiber, high-fat diet to one rich in fiber from fruits, vegetables, and whole grains, measurable improvements in colon health occurred in just 2 weeks. That's how quickly the microbiome can respond when you feed it the right way. And the benefits aren't limited to cancer prevention. Fiber-rich diets help regulate blood sugar, lower cholesterol, and support healthy weight management, all factors that indirectly protect your colon as well. Obesity and insulin resistance are both linked to higher colon cancer risk, and fiber plays a central role in mitigating these risks. By slowing digestion and promoting satiety, fiber helps prevent overeating and excessive calorie intake. At the same time, its ability to stabilize blood sugar reduces insulin spikes that can drive inflammation and cell proliferation in the colon. From a practical perspective, increasing your fiber intake doesn't require exotic ingredients or expensive supplements. It means making intentional everyday choices, swapping white bread for whole grain, replacing sugary snacks with fruit, adding a handful of beans to your salad, or starting your morning with steel-cut oats topped with berries. It's about remembering that each forkful of fiber-rich food is a gift to your microbiome, a direct investment in the strength and resilience of your colon cells. Among the most remarkable plant families in nature, the cruciferous vegetables stand out as nutritional powerhouses uniquely equipped to defend the colon. These vegetables, including broccoli, cauliflower, Brussels sprouts, kale, cabbage, bok choy, and arugula, belong to the Brassicaceae family, and their benefits extend far beyond basic nutrition. What makes them extraordinary is their high concentration of bioactive compounds known as glucosinolates. When you chew, chop, or even gently steam these vegetables, glucosinolates are broken down into active compounds such as sulforaphane, indole-3 carbinol, and phenethyl isothiocyanate. These molecules are nature's own chemical defense system, designed by the plant to ward off pests and diseases, but when we eat them, they appear to confer those same protective effects on our bodies, particularly within the colon. Sulforaphane, in particular, has been the subject of extensive research for its anti-cancer properties. It is a potent activator of a biological pathway known as NRF2, which turns on a suite of genes responsible for producing detoxification enzymes and antioxidant proteins. This pathway essentially boosts your body's internal cleanup crew, enabling it to neutralize and eliminate potential carcinogens before they can damage DNA. In the colon, this is especially important because the cells lining the intestinal wall are constantly exposed to substances from digested food, some of which may contain compounds capable of initiating cancerous changes. Sulforaphane also influences cell signaling in a way that encourages damaged cells to repair themselves or, if repair is impossible, to initiate programmed cell death. This ability to target abnormal cells while leaving healthy cells unharmed is one of the reasons cruciferous vegetables are considered a frontline dietary defense against colon cancer. Another fascinating aspect of cruciferous vegetables is their ability to modulate estrogen metabolism. While estrogen is typically associated with reproductive health, certain breakdown products of estrogen can promote cancer development in both men and women if they accumulate in excessive amounts. Indole-3 carbinol, derived from the digestion of glucobrassicin in cruciferous vegetables, helps shift estrogen metabolism toward producing less harmful byproducts. Although this is most often discussed in the context of breast and prostate cancer, it is also relevant for the colon, as hormonal balance can influence the growth environment of intestinal cells. The ability of cruciferous vegetables to influence hormone-related pathways adds another layer to their multifaceted protective role. This interaction between plant compounds and the microbiome is one of the most exciting areas of current nutritional science. suggesting that the benefits of cruciferous vegetables go beyond their nutrient profile and extend into complex biochemical partnerships within the body. The method of preparation plays a significant role in preserving the protective compounds in cruciferous vegetables. Overcooking, especially boiling for extended periods, can cause glucosinolates to leach into cooking water and degrade. Light steaming, stir-frying, or consuming them raw can maximize the preservation of sulforaphane and other beneficial compounds. Pairing cruciferous vegetables with foods rich in healthy fats, such as olive oil, avocado, or nuts, can further enhance the absorption of fat-soluble antioxidants like beta-carotene and vitamin K present in these vegetables. Even something as simple as sprinkling a small amount of mustard powder on cooked broccoli can help restore sulforaphane levels, because mustard seeds contain the enzyme myrosinase, which activates glucosinolates into their cancer-fighting forms. Epidemiological studies consistently show that populations with higher intakes of cruciferous vegetables have lower rates of various cancers, including colon cancer. In one large-scale study, individuals who consumed these vegetables at least 5 times per week had a significantly reduced risk compared to those who ate them rarely. Laboratory experiments provide clues as to why. Cancer cells exposed to sulforaphane or indole-3-carbinol show reduced proliferation, impaired ability to form new blood vessels, and increased vulnerability to immune system attack. In animal models, diets rich in cruciferous vegetables have been shown to reduce the number and size of precancerous lesions in the colon, a strong indication that these foods intervene early in the disease process. From a nutritional standpoint, cruciferous vegetables deliver far more than just their bioactive compounds. They are rich in vitamin C, vitamin K, folate, potassium, and magnesium, all of which contribute to the integrity and resilience of the colon lining. Their high fiber content works in tandem with the microbiome to produce short-chain fatty acids like butyrate, further enhancing their anti-cancer potential. The combination of direct biochemical defense, inflammation reduction, microbiome support, and nutrient density makes cruciferous vegetables one of the most complete colon-protective food groups available in nature. Polyphenols are a vast and diverse group of plant compounds that represent one of the most intriguing intersections between nutrition and disease prevention. Found in colorful fruits, vegetables, teas, spices, cocoa, nuts, and seeds, these naturally occurring chemicals give plants their vibrant pigments, distinctive flavors, and resilience against environmental stress. When we consume them, they extend those protective qualities to our own bodies. Within the context of colon health, polyphenols are emerging as one of the most effective dietary tools for reducing oxidative stress, calming chronic inflammation, and directly interfering with the biological pathways that cancer cells depend on to survive and spread. Unlike vitamins and minerals, which tend to act as individual agents, polyphenols operate as a vast, interconnected network of compounds, each with its own specific targets and mechanisms in the body. This diversity allows them to engage with multiple aspects of colon health simultaneously, from protecting DNA against free radical damage to modulating the growth of new blood vessels that would otherwise feed a developing tumor. One of the primary mechanisms through which polyphenols exert their benefits is by acting as powerful antioxidants. The cells lining your colon are constantly exposed to oxidative stress, a form of cellular wear and tear caused by unstable molecules called free radicals. These free radicals can come from environmental toxins, processed foods, chronic inflammation, or even natural metabolic processes. When left unchecked, they can damage DNA, disrupt normal cell signaling, and create the kind of cellular instability that sets the stage for cancer. Polyphenols, such as anthocyanins from blueberries, catechins from green tea, and epicatechins from dark chocolate, neutralize these free radicals before they can cause harm. This is not a one-time protective act, but an ongoing defensive process that happens every time polyphenol-rich foods are incorporated into the diet. Over time, the consistent consumption of these compounds creates an internal environment that is far less favorable for cancerous changes to take hold. Beyond their antioxidant capacity, polyphenols have a unique ability to influence the process of angiogenesis, the growth of new blood vessels. While angiogenesis is vital for wound healing and normal development, it can be hijacked by cancer cells to secure a steady supply of nutrients and oxygen, allowing them to expand rapidly. Certain polyphenols, particularly those found in green tea, EGCG, and pomegranate elagitannins, have been shown to inhibit this abnormal blood vessel growth. In the context of colon cancer, cutting off a tumor's ability to feed itself is a critical step in halting its progression. This anti-angiogenic property is especially valuable because it targets the disease in a way that does not rely on directly killing cells, but rather on starving them into dormancy, reducing the likelihood of collateral damage to healthy tissues. Inflammation is another key factor in the development of colon cancer, and polyphenols have been repeatedly demonstrated to suppress inflammatory pathways at the molecular level. Chronic inflammation can result from poor diet, infections, autoimmune reactions, or long-standing gastrointestinal conditions, and it creates a biochemical environment rich in signals that promote cancer growth. Polyphenols like curcumin from turmeric, resveratrol from grapes, and quercetin from onions can downregulate inflammatory mediators such as COX-2 enzymes and pro-inflammatory cytokines. By reducing these molecular signals, polyphenols not only decrease the overall inflammatory burden in the colon, but also interrupt the feedback loops that cancer cells use to promote their own survival and spread. One of the most fascinating areas of research is the interaction between polyphenols and the gut microbiome. Many polyphenols are not fully absorbed in the upper digestive tract. Instead, they travel to the colon, where they interact directly with gut bacteria. This interaction is not passive. Bacteria can metabolize polyphenols into smaller, more bioactive compounds that have even greater protective effects. For example, elagitannins from berries and pomegranates are converted by gut bacteria into urolithins, compounds that have been shown to reduce inflammation and induce cancer cell death in colon tissue. In turn, polyphenols can promote the growth of beneficial bacterial strains such as lactobacillus and bifidobacterium, creating a more balanced and resilient gut ecosystem. This bidirectional relationship suggests that a polyphenol-rich diet not only delivers direct biochemical benefits, but also shapes the microbiome in ways that further enhance colon protection. The variety of polyphenols available in the diet also allows for a broad-spectrum approach to colon health. Flavonoids, still beans, phenolic acids, and lignans each have distinct molecular targets and benefits. Flavonoids, found abundantly in citrus fruits, parsley, and tea, are particularly adept at scavenging free radicals and stabilizing cell membranes. Still beans like resveratrol, concentrated in red grapes and berries, are well known for their anti-inflammatory and anti-angiogenic properties. Phenolic acids, found in coffee, whole grains, and certain berries, have been shown to modulate gene expression related to detoxification enzymes in colon cells. Lignans, present in flax seeds and sesame seeds, not only act as antioxidants, but also influence hormone metabolism in ways that may further reduce colon cancer risk. This diversity means that no single food holds the monopoly on polyphenol benefits. Instead, the goal is to consume a wide variety of plant-based foods to ensure comprehensive coverage. From a dietary perspective, increasing polyphenol intake is as much about variety as it is about quantity. Deeply pigmented fruits such as blueberries, blackberries, cherries, and plums offer concentrated doses of anthocyanins. Green and white teas provide catechins, while black tea supplies theaflavins. Dark chocolate with a high cocoa content offers flavonols, and spices like turmeric, cloves, and cinnamon. Add curcumin and other potent compounds to the mix. Herbs such as rosemary, thyme, and oregano are not only flavorful additions to meals, but also concentrated sources of polyphenols. Whole grains, nuts, and seeds contribute additional phenolic acids and lignans, rounding out the spectrum. By strategically including these foods across meals and snacks, it is possible to create a constant flow of polyphenols through the digestive system, maintaining steady protective pressure against the processes that lead to colon cancer. For decades, the word fat carried a negative connotation associated with weight gain, heart disease, and poor health outcomes. Yet, modern nutritional science has completely reframed our understanding, revealing that not all fats are created equal. While trans fats and excessive saturated fats can harm the body, certain types of fats are essential for optimal cellular function, hormone regulation, and disease prevention. Among these, omega-3 fatty acids stand out as particularly important in protecting colon health and reducing cancer risk. Found abundantly in fatty fish such as salmon, mackerel, sardines, and anchovies, as well as in plant-based sources like flax seeds, chia seeds, hemp seeds, and walnuts, omega-3s possess potent anti-inflammatory, immune-modulating, and cell-protective properties that make them a crucial dietary component in colon cancer prevention strategies. The anti-inflammatory effects of omega-3 fatty acids are among their most powerful benefits. Chronic inflammation in the colon creates a microenvironment that encourages abnormal cell growth, DNA damage, and the development of precancerous polyps. This inflammatory state often results from a combination of dietary imbalances, high consumption of refined carbohydrates and processed meats, and low intake of plant-based nutrients. Omega-3s help disrupt this cycle by serving as building blocks for anti-inflammatory molecules called resolvins and protectins. These compounds actively resolve inflammation rather than simply suppressing it, restoring balance in the colon's immune environment. This is particularly important for individuals with inflammatory bowel diseases like ulcerative colitis and Crohn's disease, as both conditions dramatically increase colon cancer risk when inflammation remains uncontrolled. By integrating omega-3 rich foods into the diet, it becomes possible to reduce inflammatory signaling pathways, calm irritation in the colon lining, and create conditions less favorable to cancer progression. Beyond reducing inflammation, healthy fats play a direct role in the structure and function of cell membranes. Every cell in your body, including those lining the colon, is surrounded by a lipid bilayer, a membrane made largely of fats. The types of fats you consume directly influence the fluidity, stability, and functionality of these membranes. Diets rich in omega-3s and monounsaturated fats, such as those found in olive oil, avocado, and almonds, result in membranes that are more flexible and responsive, allowing cells to communicate more effectively, transport nutrients efficiently, and respond to damage in a controlled manner. In contrast, diets dominated by unhealthy fats can produce rigid, less functional cell membranes that are more susceptible to oxidative stress and inflammation, both of which are linked to cancer development. Healthy fats therefore do more than just provide energy. They fundamentally shape the physical and biochemical environment in which cells live and function. Another critical way in which omega-3s protect the colon is by influencing gene expression. These fatty acids can interact with nuclear receptors that regulate the transcription of genes related to inflammation, cell proliferation, and apoptosis. In simple terms, omega-3s can help switch off genes that promote tumor growth while switching on genes that support normal cell cycle regulation and programmed cell death. This dual effect makes them a unique dietary tool. They can simultaneously reduce the signals that drive cancer and enhance the body's ability to eliminate damaged cells before they can become a threat. Laboratory studies have shown that colon cells exposed to higher concentrations of omega-3s demonstrate reduced proliferation rates and increased sensitivity to apoptosis, even in the presence of pro-cancerous stimuli. Healthy fats also work in synergy with other protective nutrients in the diet. For example, many of the bioactive compounds found in cruciferous vegetables, polyphenol-rich foods, and certain spices are fat-soluble, meaning they require the presence of dietary fats for optimal absorption. Sulforaphane from broccoli, curcumin from turmeric, and carotenoids from carrots and leafy greens all become more bioavailable when consumed with a source of healthy fat. This means that adding avocado to a spinach salad, drizzling olive oil over roasted Brussels sprouts, or pairing salmon with a side of sautéed kale not only enhances flavor, but also maximizes the nutritional benefits of the meal. In the context of colon health, this synergy can significantly amplify the protective effects of an anti-cancer diet. Emerging research is also exploring the impact of the omega-6 to omega-3 ratio on inflammation and cancer risk. While omega-6 fatty acids found in many vegetable oils like corn, soybean, and sunflower oil are essential in small amounts, excessive intake relative to omega-3s can promote a pro-inflammatory environment. Historically, human diets maintained a balance between these two types of fats, but modern eating patterns have skewed heavily toward omega-6s, often at a ratio of 15.1 or higher, compared to the more optimal 2.1 or 3.1. This imbalance can fuel chronic inflammation, undermining the body's natural defenses against cancer. Restoring a healthier balance by reducing processed vegetable oils and increasing omega-3 rich foods is a practical, evidence-based step toward lowering colon cancer risk. Monounsaturated fats, while less discussed than omega-3s, also play a meaningful role in colon health. Found in abundance in extra virgin olive oil, macadamia nuts, and avocados, these fats are resistant to oxidation and have been shown to support healthy cholesterol levels, reduce oxidative stress, and improve insulin sensitivity. Since insulin resistance and metabolic syndrome are associated with increased colon cancer risk, including monounsaturated fats in the diet can provide another layer of protection. The Mediterranean diet, which is rich in both omega-3 and monounsaturated fats, has been extensively studied and repeatedly linked to lower rates of cancer, including colorectal cancer, in part because of this balanced fat profile combined with an abundance of plant-based foods. In addition to their systemic effects, healthy fats influence bile acid metabolism in the digestive system. When dietary fats are digested, the liver releases bile acids to emulsify them. Certain bile acids, when present in high concentrations and not adequately bound by dietary fiber, can become harmful to the colon lining, promoting inflammation and cellular stress. Omega-3s appear to help modulate bile acid composition in a way that is less irritating to the colon, while fiber intake, often paired with these fats in whole foods, helps carry excess bile acids out of the body. This partnership between healthy fats and fiber-rich foods creates a digestive environment that is cleaner, less inflammatory, and less conducive to cancer development. Fermented foods have been part of human diets for thousands of years, long before refrigeration or modern preservation methods. Across cultures and continents, fermentation emerged not only as a way to extend the shelf life of food, but also as a means of enhancing flavor, texture, and nutritional value. What science has now confirmed is that fermentation also transforms ordinary ingredients into powerful, functional foods that can play a significant role in protecting the colon. Fermentation relies on the action of beneficial microorganisms, bacteria, yeasts, and sometimes fungi that metabolize sugars and other compounds in food to produce organic acids, enzymes, and bioactive molecules. When consumed, these foods deliver living microorganisms known as probiotics, along with their fermentation byproducts, directly into the gut. This influx of beneficial microbes and metabolites can profoundly influence the health of the colon by shaping the composition of the gut microbiome, reducing harmful compounds, and strengthening the intestinal barrier. The human gut is home to trillions of microorganisms that form a dynamic ecosystem critical for digestion, immune function, and disease resistance. However, this delicate balance can be easily disrupted by poor diet, antibiotics, chronic stress, and environmental toxins. An imbalanced microbiome, often referred to as dysbiosis, is characterized by a decrease in beneficial bacteria and an overgrowth of harmful species, creating an environment more prone to inflammation, increased permeability of the intestinal lining, and the production of carcinogenic compounds. Fermented foods act as reinforcements for the microbiome, introducing beneficial bacteria such as lactobacillus, bifidobacterium, and Saccharomyces, which compete with and displace harmful bacteria, reestablishing microbial diversity. A diverse microbiome is better equipped to produce short-chain fatty acids like butyrate, which are essential for nourishing colon cells, regulating immune responses, and suppressing tumor formation. The metabolites produced during fermentation also have direct anti-cancer properties. For example, lactic acid bacteria found in yogurt, kefir, kimchi, and sauerkraut can produce compounds that inhibit the activity of enzymes such as beta-glucuronidase, which are involved in the activation of carcinogens within the colon. By reducing these enzyme activities, fermented foods help lower the burden of harmful substances that could damage the DNA of colon cells. Furthermore, certain probiotic strains can bind to and neutralize potential carcinogens before they have a chance to interact with the colon lining. This detoxifying capacity of probiotics is a key reason why fermented foods are being increasingly studied as part of comprehensive strategies for colon cancer prevention. The immune-modulating effects of fermented foods are another dimension of their protective power. The gut is the largest immune organ in the body, with a vast network of immune cells stationed along the intestinal lining. Probiotic bacteria from fermented foods interact with these immune cells, training them to mount efficient responses against harmful invaders while avoiding excessive chronic inflammation that can damage tissue and foster cancer development. Some probiotic strains stimulate the production of anti-inflammatory cytokines, while others enhance the activity of natural killer cells and macrophages, which can identify and destroy early-stage cancer cells. This fine-tuned immune regulation is essential in the colon, where the immune system must constantly balance the need to defend against threats with the need to tolerate harmless food particles and resident microbes. Fermented foods also contribute to strengthening the intestinal barrier, the thin but critical layer of cells and mucus that separates the contents of the gut from the bloodstream. When this barrier becomes compromised, a condition often called leaky gut, harmful substances can pass through and trigger systemic inflammation, which is linked to increased cancer risk. Probiotics and their fermentation byproducts, such as lactic acid and certain peptides, can enhance the production of tight junction proteins that keep intestinal cells closely bound together, making the barrier more resilient. A strong barrier not only reduces inflammation, but also limits the entry of potential carcinogens into the deeper layers of the intestinal wall. Another intriguing aspect of fermented foods is their ability to transform the nutritional profile of the raw ingredients from which they are made. During fermentation, certain nutrients become more bioavailable. For instance, fermentation can increase the levels of B vitamins, vitamin K2, and certain antioxidants in foods. It can also break down anti-nutrients like phytates, which normally bind to minerals and inhibit their absorption. In the case of colon health, the improved availability of minerals such as calcium and magnesium supports cellular integrity and proper function in the intestinal lining. Additionally, some fermented foods produce unique bioactive peptides, short chains of amino acids with biological activity that have been shown to exhibit antioxidant, anti-inflammatory, and even anti-proliferative effects against cancer cells. The variety of fermented foods available across cultures offers countless options for incorporating them into the diet. Yogurt and kefir are perhaps the most familiar, providing dairy-based sources of probiotics along with calcium and protein. For those avoiding dairy, plant-based alternatives such as fermented coconut yogurt or almond kefir can offer similar benefits. Sauerkraut and kimchi deliver live bacterial cultures alongside fiber and phytochemicals from cabbage and other vegetables, which act synergistically to nourish the microbiome. Miso, tempeh, and natto, made from fermented soybeans, provide probiotics along with isoflavones, plant compounds with additional anti-cancer potential. Even fermented teas like kombucha contribute beneficial acids and antioxidants, although their probiotic content can vary. Choosing minimally processed, unpasteurized versions of these foods ensures that the live microbes remain intact and active when consumed. The frequency and consistency of fermented food consumption matter greatly. While a single serving may provide a temporary boost of beneficial bacteria, regular inclusion in the diet helps maintain a stable population of probiotics in the gut. This steady presence ensures ongoing production of beneficial metabolites, continued immune support, and a lasting influence on microbiome composition. Pairing fermented foods with prebiotic-rich foods, those high in fibers that serve as food for probiotics, can further enhance their effectiveness. Examples include combining yogurt with berries, sauerkraut with a whole grain sandwich, or kimchi alongside stir-fried vegetables. This symbiotic pairing ensures that beneficial bacteria have the fuel they need to thrive and multiply once inside the colon, amplifying their protective impact. Spices have been valued for centuries, not only for their ability to enhance flavor, but also for their medicinal properties. Long before modern science could explain their effects, ancient healing traditions like Ayurveda and traditional Chinese medicine recognized certain spices as powerful agents for maintaining health and preventing disease. Today, research confirms that many culinary spices contain concentrated levels of bioactive compounds capable of influencing human biology at the molecular level. When it comes to colon cancer prevention, these compounds can act on multiple fronts, reducing inflammation, neutralizing carcinogens, altering cell signaling, and even triggering cancer cell death. Because spices are used in small quantities yet deliver highly potent phytochemicals, they can be thought of as concentrated nutritional tools, easy to incorporate into daily meals without adding excessive calories, while offering profound protective benefits. These compounds can also directly interfere with cancer cell replication, disrupt tumor cell metabolism, and stimulate the immune system to identify and destroy abnormal cells. Epidemiological studies have found that higher garlic consumption correlates with a lower risk of colorectal cancer, and some clinical research suggests that aged garlic extract may help reduce the recurrence of precancerous polyps. The way garlic is prepared matters, crushing or chopping fresh cloves and allowing them to rest for several minutes before cooking increases the formation of allicin, maximizing its health benefits. Ginger, a root with a long history in both culinary and medicinal use, offers a different but complementary set of protective effects. It contains bioactive compounds like gingerols, shogaols, and paridols, which have been shown to reduce oxidative stress, suppress inflammation, and inhibit angiogenesis, the formation of new blood vessels that tumors need to grow. Ginger's anti-inflammatory action is partly due to its ability to inhibit the same COX-2 enzyme targeted by curcumin, but through slightly different mechanisms, making the two spices a powerful combination when used together. Animal studies have demonstrated that ginger extract can reduce the number and size of colon tumors, while human trials indicate it can lower markers of inflammation in the colon lining, particularly in individuals at elevated risk for colorectal cancer. Cinnamon, a spice derived from the bark of cinnamomum trees, provides another example of a flavor-rich ingredient with measurable anti-cancer potential. It contains cinnamaldehyde and procyanidins, which have been observed to modulate signaling pathways involved in cell proliferation and apoptosis. Cinnamon also has the ability to improve insulin sensitivity and regulate blood sugar levels, an important benefit given that insulin resistance and high circulating insulin levels are associated with increased colon cancer risk. By helping maintain stable glucose metabolism, cinnamon indirectly reduces one of the metabolic pressures that can contribute to abnormal cell growth in the colon. Additionally, its polyphenol content adds to its antioxidant capacity, further protecting colon cells from oxidative damage. Cloves, though often used sparingly due to their intense flavor, are among the richest natural sources of antioxidants, particularly eugenol. Eugenol has been shown to inhibit tumor cell proliferation, induce apoptosis, and suppress inflammation in various cancer models, including those involving the colon. Its high antioxidant activity also helps prevent lipid peroxidation, a process where free radicals damage cell membranes, creating conditions favorable for cancer initiation. Cloves, like many spices, are best used freshly ground to preserve their volatile compounds, which can dissipate over time when exposed to air and light. The synergistic potential of spices is another important consideration. While each spice offers unique bioactive compounds, their combined effects can be greater than the sum of their parts. For example, turmeric's curcumin and black pepper's piperine enhance each other's bioavailability, while ginger and garlic together provide a broader spectrum of anti-inflammatory and detoxifying compounds. Traditional spice blends such as curry powder, garam masala, and ras el hanout often combine several protective spices in one recipe, offering not just culinary complexity, but also an integrated set of anti-cancer phytochemicals. These blends, especially when made from fresh, high-quality ingredients, can become powerful tools for supporting colon health when used regularly in cooking. Incorporating these spices into the daily diet does not require major changes in eating habits. Small, consistent additions can provide cumulative benefits over time. Turmeric can be stirred into soups, stews, or smoothies. Garlic can be incorporated into salad dressings or sautéed vegetables. Ginger can be grated into teas, stir-fries, or marinades. Cinnamon can be sprinkled over oatmeal, yogurt, or roasted vegetables. And cloves can add depth to spice rubs, baked goods, or warm beverages. These everyday uses make it possible to create a steady intake of bioactive compounds without relying on large supplemental doses, which sometimes lack the synergistic components found in whole spices. By consistently exposing the colon to this wide range of protective phytochemicals, spices offer a practical, flavorful, and scientifically validated approach to disrupting the pathways that lead to cancer cell growth and survival.